Hello. Oh, I am in a completely different time zone, so it took me a minute to get everything sort of sorted and worked out. And whew, I was also offline all day yesterday as I traveled. And when I get back, it's like the flood of stuff and information and just finding the right balance, everything. So I am really happy to be here. Um, Super excited that I made it. For those of you that know where I am, like this will give you the reference of where I am and such. And yeah, so I had to also sort out the paper because I was very torn between two different papers that I wanted to go through, especially because one of them is slightly well, actually, they both are kind of technical, but I think that they provide a really great abstraction of models that we can look at. And you know that here we have been going through a lot of the type of commentary that's more philosophical in nature. And here we're going to get into another paper that's more biological in nature. And then how could we look at that biology to extrapolate models that we can work with? So this is really an important aspect that I think I don't emphasize enough, and I want to start emphasizing more, which is that plants as models, plants as mentors, and plants as partners, and plants also as measure, right? Measure in the sense of helping us understand like in how much we should do something or not do something to help us give us different types of ways of interacting and working with plants. And I mean, this happens with all persons, right? So even human people, you have those people that you look at as kind of models that you think about and you watch from afar and you say, oh, I would really like to look at that way that they do things. And then you have also humans that you look at from the perspective of mentors and you have a relationship with, and they teach you from their experience. And then you also have these people that you're collaborating with, that you're actually kind of at a partnership level and as a same level. So I think it's really important to help, and some that are actually your moral compass, right? That help you really understand how your own moral beliefs and thoughts and processes. I'm kind of bothered right now because this computer I didn't think about this beforehand. I usually prompt it up on a really beautiful book that's way too far right now for me to go and get. But I want to that did pretty much nothing. I want to raise my computer a little bit because I don't like it that you're looking up at me. So I'm going to use my mother's Italian book. This is my mother learning Italian in 10 minutes a day. She has a lot of fun with it. So I'm going to use her book and I'm going to prompt you up a little bit more and see if this kind of gives me a better angle. I don't want you just looking up at me. I think it's crazy. Anyway, so she has all these other things to them. So pardon me while you're being a part of my process here, which is kind of a crap process, but it's all good. It works. Okay, so slightly angled, not in a way that I feel super comfortable with, but where it's going to have to do for right now. Okay. No, I'm going to take off this. Sorry. Thank you for being super patient with me as I work through all of these types of things. Okay, this is better. This is the best I'm going to get to right now. All right, let's move forward and let's go into this paper. So happy Friday for everyone. I hope that wherever you are, you're doing really well. Thank you for watching. If you're watching this on a replay, awesome. Please leave any comments or questions, concerns, thoughts, anything you might have so that we can address this, or you can just come into the Naturally Conscious community and, watch us live. So here is what we're going to talk about. We are going to talk about epigenetic memory as a basis for intelligent plant behavior in clonal plants. So let's give some definitions before I share my screen because it's important for us to understand. Epigenetic, for those of you that don't know what they mean by what we're talking about when we're talking about epigenetic, we are talking about things that are relating to changes, specifically things that you can inherit or characteristics of an organism, it can also be of a cell or whatever, that result from altering the expression of a gene. In other words, these do not involve changing the DNA sequence itself, like how many genes you have or don't have, but more of how is it that gene expresses itself. So how is it that you're moving? This really is something connected oftentimes to the whole idea of nurture versus nature, right? When we talk about nature, you're talking about what were you born with? What is it that is your physiological perspective of what you are born with From a spiritual perspective, I would also add, like, if you're talking about what is the structure of your soul, what are the inherent traits and personalities that you come into being with, but that's for the audience that really, is thinking about this beyond, and the fact that we are both a physical as well as a spiritual body. So what are, regardless of whether you're talking about physically or spiritually, what are all the elements that you come into being with? Right? That is my nature perspective. And then my nurture perspective is the idea of how do those express themselves by the way that we are, by what we live in. How are we nurtured? What are the things? What does our environment give us? So epigenetic changes are oftentimes also reversible. This is the things like when the recent article in Sprout discussions around the idea of my personalities and how do my personalities kind of take hold? How do they show? How do they change my body? Nothing changes my DNA itself, but it changes how my DNA is read. You can think of it in that way. And this is the exact same thing that we're talking about relating to even from a spiritual perspective. This doesn't change the composition of my soul, the composition of my attractor with all my personalities, but instead how those personalities interact with one another and express themselves through me. So this is what we're talking about when we're talking about epigenetic changes. And in this particular paper that we're talking about epigenetic memory, so the idea of remembering what these changes are or how these changes have been experienced as a basis for the intelligence, for the behavior. So it provides information that allows for an intelligent movement or an intelligent response, I guess that's the word I'm looking for, of clonal plants. So in the case of clonal plants, we're talking about plants that reproduce themselves by creating clones of themselves. So things that have, for example, if we talk about aspen trees, right? Pando, if you read my recent article about Pando is a clonal, is an aspen, a quaking aspen trees. And aspen trees represent, they reproduce clonal. So this is there's lots of plants that produce that are able to reproduce themselves by producing a kind of, I think they're called ramets. So they produce a sort of ramet, or that looks and is capable of independent growth and even of of dispersal, of moving on into other directions. And that ramet that gets created, is a way for them to reproduce exactly. So the DNA sequences are the same because technically from a genetic perspective, all of what you're seeing you look at something like Pando, all this forest filled of quaking aspens, all are actually the same tree from a genetic perspective. So that DNA, that phenotype, rather than the expression, the epigenetic expression is different because they are in different areas. So something as large as Pando, which is this giant forest of quaking aspens, the ones that are maybe more on the south side and are getting more sun will grow in certain ways and will have certain kinds of genetic expressions, where those that are growing more in the north or maybe more in a shaded area will have a different kind of genetic expression, even though all of their genetics are absolutely the same because they are clones of one another. The way they express themselves is very much connected to the area in which they grow. So hopefully that makes sense. I'm trying to think of other plants that tend to be clonal. There's lots of different, I guess you could look at things like sweet potatoes are specific kind of potatoes are also clonal. There's lots of different types of clonal plants. I'm looking at a search right now to see if there's any specific ones that I could think about that you might kind of identify with really easily. Certain fungi are clonal, and this is a really bad article that I just clicked on because it doesn't give me an actual list of clonal plants. So it gives me something completely different. But anyways, you get the idea. So this is where we're talking about. This is the basis and the foundation upon which we're going to talk about right now. So let me share my screen with you so that we have the paper, as we do every week. I feel like it's still really dark. I set up a light, but I feel like I have very little light here. I have to set up my whole setup. Today has been a little bit crazy as I get ready for everything. Okay. Uh-oh. Oh, there it is. Okay. So let's move on. Here we are with our paper. Our paper is called, again, Epigenetic Memory as a Basis for Intelligent Behavior in Clonal Plants. I only have one screen right now, so it's going to be a little bit trickier than it usually is. Okay, so this is a paper that has been written by Vit Latzel, Alexandra P. Redina Gonzalez, and Alejandra Rendina Gonzalez, and Jonathan Rosenthal. This is from the Czech Academy of Sciences, which is kind of interesting. We haven't seen this, and also the Ecological Research Institute in New York. So these are the two. As always, in the show notes, I will include all the information about this paper itself and where you can download it if you're interested in. So let's just get into it. Environmentally induced epigenetic changes enables plants to remember past environmental interactions. If this memory capability is exploited to prepare plants for future challenges, it can also provide a basis for highly sophisticated behavior considered intelligent by some. Seriously. Considered intelligent by some. We are still. This paper, by the way, I didn't tell you the year because I know we always talk about it. This paper was submitted in 2016 March, and it was published in August of 2016. So I can understand a little bit why they are saying, considered intelligent by some, because this is kind of the tail end of the bigger discussion around intelligence in plants. And of course, we're still at the part where intelligence is being quantified, and qualified. In other words, it's not just intelligence, period, but it's more like intelligence based on blah, 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 blah. And there's lots of qualifiers that happen in there. So we're still in that era, but we're near the tail end of it. Now, not everybody is up to speed. And by this point, there are a number of people who are very comfortable in talking about plants as intelligent. But, this is young researchers that probably didn't want to get themselves in trouble, so they didn't want to mince their words too much. Anyway, it can, let's see. Against the backdrop of an overview of plant intelligence, we hypothesize, one, that the capability of plants to engage in such intelligent behavior increases with the additional level of complexity afforded by clonality. So in this case, is the fact of, remember that by afforded, we mean what is that is offered to you just because that thing is that way. So an easy affordance that I use the example of all the time is if you see a door and the door has no handle, and you're supposed to go through this door and it has absolutely no handle, your first inclination is to push the door. That is an affordance. In other words, there's no sign, there's no necessity for you to add anything extra. Just the fact that the door has no handle on it implies to you that it's probably a push, as opposed to if the door had a really nice ornate handle, you would probably pull the door. So these are called affordances. Affordances is a really important aspect of sociology, of understanding how our mind and our cultures teach us to traditionally interpret things. And this can help us when we are creating and designing a lot. So in this case, the affordances, the fact that they're a clone, which means a DNA copy, means that the behaviors that they express are shared from one to the other. They don't have to be relearned because if it's part of their DNA sequencing, or in this case, it's part of what is being inherited from one to the other as the new clone grows. And this is the interesting part, is they're testing whether or not epigenetic changes, so nature or environmentally accelerated changes actually get inherited from one to the other. And does that mean that the clone who has not just the DNA that allows them to do something, but also the epigenetic expression of it, because it's a clone, rather than having to relearn the behavior and express the gene in that way, inherently have that as a starting point, which I think is a really fascinating conversation point from the perspective of, I think about my own dumb and hurriedness. We have one of the letters that came to us from Falco, our spiritual founder, in the beyond, that arrived, had a really important statement that a lot of us have not only appreciated, but come to really understand, which is, he said to us, the next generation doesn't have to start from the beginning. They should start from where you left off. Which means that they shouldn't have to go through all the struggles to learn things that we all had to struggle through and learn through in the beginning. Instead, they should start from the place. We should start helping them to move from the place where we are today to the future. We see this also in Dumb and Hurry and art. Dominant art is inspired from dominant art in the perspective of you should start with dominant art as a form, like, or any other kind of type, but you should start with the greatest expression of dominant art and build new art from that point on going forward. So this is kind of testing out that same ability, which is, do clonal plants pass off what they have learned to the next generation, even though their DNA is identical, but they also pass on kind of what they've experienced and what they've learned from in relating to their environment and to the way that they've lived, and then pass that on so that the next generation has a kind of leg up, is already actualized to what is or they're integrated into whatever that current environment is, rather than starting with a clean DNA slate. So hopefully I didn't make that too long, but I was trying to make it clear. 2, that more faithful inheritance of epigenetic information in clonal plants, in conjunction with information exchange and coordination between connected ramets, is likely to enable especially advanced intelligent behavior in this group. So this is interesting too, because it's the idea of if the plant is able to inherit and really be faithful, so really follow through with what is that type of lessons that are learned, that complexity, that level of evolution that has been that they arrived at, information with exchanging information between the ramets. So kind of like the equivalent of, well, why did you do this? So that I better understood not just how you're expressing it, but also how you got to that expression. Would that lead to more intelligent behavior because you're already starting from a place of awareness that is not a blind spot? Like, I don't just start out dull. I don't just start out at a place where I have to gather everything up myself. I think this is really interesting. We therefore further hypothesize that this behavior provides ecological and evolutionary advantages to clonal plants, possibly explaining, at least in part, their widespread success. I mean, Pando is a great example of a really widespread success, but also things like potatoes and lots of other crops that we're using, ginger, these types of things. Finally, we suggest avenues of inquiry to enable assessing intelligent behavior and the role of epigenetic memory in clonal species. All right, let's get into it. Okay, so introduction. Although it has historical antecedents in remarks by Darwin, the most recent and not universally held characterization of plants as intelligent or, excuse me. Why does it jump? Hold on, I'm trying to make a nice level of, all right, there we go, that's better. Held characterization of plants as intelligent organisms is largely attributable to a series of publications by Trawawas. We've talked about Trawawas so many times. Indeed, Trawawas' arguments for plant intelligence, coupled with counter arguments, which we've gone through all of these papers, to his viewpoint, capture the overall discussion on this subject quite well. In the present paper, we propose investigation of a system that is likely to especially strongly display those behaviors that Trawawas and we would characterize as intelligent. Moreover, this system is particularly well suited to address the arguments put forth to dispute plant intelligence. To address those arguments. Interesting. Finally, we hypothesize that to this extent, the intelligence is manifested in the behavior of the focal organism, clonal plants. It may, at least in part, explain the ecological and evolutionary success of their life strategy. Sorry, I really have to drink today because I have drank so little over the last 24 hours, especially compared to what I normally did. And so it's like, if I don't drink something, I'm going to be parched. At least the temperature here is very much in favor of taking good care of my throat. With its nice warmth. Okay, first, a brief overview of Trawawa's arguments and the proffered rebuttals is in order. As he noted, the traditionally prevailing presumption that plants lack intelligence is largely due to methodological difficulties and biases in applying to plants concepts originally developed in the context of animals. We know this, we've talked about this so many times. In particular, because the traditional zoocentric measure of behavior has been in terms of widespread movement plants, in particular, because the traditional, zoocentric measure of behavior has been in terms of movement, Plants' sessile lifestyle, rooted into the ground, and different reactions, time scale, has probably helped create the widespread view of plants as passive and therefore incapable of behaving. It's a very, very nice way of putting it, let alone exhibiting intelligence. Similarly, Trowavas noted that because many plant responses to environmental conditions are manifested in terms of growth or through such cellular level processes as changes in turgor pressure, it is tempting to dismiss these responses as, my page changed, as developmental or physiological processes rather than, move away, rather than behavior. So in other words, the fact that the way that a plant moves in some ways is based on the change in the pressure inside of their stems. That's what the turgor pressure is talking about, the way that the sides and the pressure between the phylum and the zoom going through and things of this nature. It is tempting to dismiss that as something that's a physiological process, which I think is so hysterical, because obviously if I was to not think about the human brain and I was just to look at an arm, for example, or fingers, of course, the way that they move is based on the pressure of the contractions and all these types of aspects. But only because I can express that I'm thinking about movement and then I move, do you think of that as intelligence behavior, right? You don't just think of it as a simple philosophical, like a instinct. I tell you I'm going to move my finger, then I move my finger. We cannot read plant minds, at least most of the scientists cannot read, let me say this better. The general scientific community cannot read a plant mind in a way that is scientifically rigorous, different than the way that we communicate with plants. So therefore, they can't fathom the idea. It doesn't even come into their minds that it's possible that the plant is thinking to the body, hey, move your trunk, and then spends the next, I don't know, whatever, 12 hours slowly moving the stem or whatever, the trunk, and the next 12 years slowly moving that trunk. It's like impossible to conceptualize that that's happening, but you can't negate it either. You have no way of negating it, but yet we struggle with that. It's amazing to me. Anyway, it is through such directed growth, however, given their sessile nature, it is through such directed growth that plants can explore and preferentially exploit favorable environments, akin to the movement shown by animals. Therefore, Trowavas employed Stenhouse's objective definition of intelligence as adaptively variable behavior within the lifetime of an individual, because it is not a priori inapplicable to plants. However, he did so with the understanding that behavior be limited to responses on the level of the whole plant, i.e., excluding direct responses of a single organ or tissue to the environment in which they were interacting. All right. See, remember I was talking about how we're still at the stage where many of the definitions around intelligence have to have qualifiers? Example, qualifier. Along with many examples of intelligence so defined, he described various plant capabilities that make such intelligence possible, including plant learning abilities and communication and decision-making capabilities. Trowava's characterization of plants as intelligence was disputed on multiple bases, the most salient of which can be distilled down to the contentions that the concept of the individual is largely inapplicable to plants, as they are not only modular, but organs can exist on their own. So the idea of the whole, goes to the conversation we had last week about when, and actually I have to say, a little side note here for a second, a super side note. I'm gonna stop the sharing for a second. So here's a side note. So last week we talked a lot about the whole idea of like the individual and the conversations that Matthew Hall was having around, the individual and such like that. And it got me thinking. So last Saturday, if you remember, unfortunately not with me right now, but those of you who know and can kind of go back through the naturally conscious community conversations, if you look for this, you've heard me talk a lot about no-name Dracaena, right? So Dracaena, this mother-in-law's tongue or snake plant that I have been working with in relation to movement, of understanding a whole series of information through movement rather than words. So no-name Dracaena is in and no name because the Dracaena has told me that they don't want to have a name because no-name Dracaena doesn't want me to be stuck in words and things of that nature. So no name. So no-name Dracaena is in a teapot. this is a teapot I bought a long time ago. The lid of the teapot broke, and so therefore I turned the teapot into a planter. And I love this planter. And no-name Dracaena decided to put up a whole new, I don't know what they would be called because it's not really a stem the way this plant, a whole new leaf cluster. And being a plant that's clonal, actually very adapt for this conversation. So put up this new stem. And of course, the teapot only has a small opening on the top. So that one was kind of getting stuck. And so it was interesting because rather than all the others, the leaves come in and each leaf sort of grows really long and then the plant stays in this sort of bushy size, this particular one came up really fast. And in order to adapt to the fact that it had very little room, rather than putting out lots of leaves that sort of slowly opened and has like still that cylindrical shape that mother-in-law's tongues or slake plants have, but having it in a little bit more of space, put it up a very tight one that went really tall. So super tall and really tight. So the other day I got this message from no-name Dracaena that it was time to move this one piece because this one piece is now like this one stem cluster or ramet, I guess it would be, is now getting to like, it doesn't fit in this pot. So I divided key into two. And since then I did not have time before I left to have a dance session or a movement session or a meditation session or anything in order to ask Key about this particular information. But I am now freaking out in some ways in a good way, because is this still all no-name Dracaena, or is it now no-name Dracaena? Is one of them no-name Dracaena and the other one is somebody else? And if so, which one of the two is no-name Dracaena? This is so interesting. Even though I've divided plants before, I don't have a tendency to try to propagate my plants unless I get a message directly from the plant saying, please propagate me. I do prune a little bit here and there, or I try to get bigger pots. I try to very much listen to whatever it is that the plant wants and is feasible. We try to have a conversation about what's feasible within my home. I'm talking obviously about plants that live with me. So this particular plant was very clear about, no, I want to be divided. And I divided without thinking, I just listening in this particular case and thought, oh yeah, and I actually had two pots, which is very rare. I don't, whatever. Anyways, but now do I have two no-name Dracaenas or is one no-name Dracaena? This is going to be something that I'm going to be thinking about a lot. I'm going to see if I can connect remotely to no-name Dracaena while I'm gone. If not, more coming soon. I'm going to be having a conversation with no-name Dracaena about this. So just so you know. Anyways, that was my long aside. It was a very, very long aside. But I found it a fascinating aside. I don't know about you, but to me, it was absolutely fascinating. All right, we're going to go back to the paper. But if you have any ideas, like if you've had a plant that you've divided, and what do you find? Are they both the same plant? Are they now taking on different characteristics? Does one for you feel like the original, where another one feels? Because in this particular case, both of these pieces are very different in shape, but they are both very much were present in, no-name Dracaena. So to me, it's not like easy. And I have to admit, I can't, because one of them is in the same position that they were before, it feels like that's no-name Dracaena, but I'm not really sure. And I think that that's just a mental bias. And anyway, so this is going to be a fun experimentation. I'm really looking forward to this experiments with this plant. Okay. Coming back to the plants. Let's see, where were we? Sorry, I totally went off on a thing. Okay. So they don't respond as individuals. That's what we were talking about. And that memory, such as it exists in plants, is similarly localized, i.e. there's no repository of plant-wide memories analogous to a brain, and simply consists of linear sequences of developmental experiences. And that, unsurprisingly, given these limitations, plants do not make meaningful choices. This was all what was coming through as an argument against intelligence. So basically, they're not individuals. Because there is no central repository, you can't even consider the individual pieces to all have the same information, because there's no one place where that information is being stored and they're all coming to. And so therefore there is no really memory analogous, and there's just a bunch of experiences that they have. And it's sort of like they have this experience and then they forget these experiences. So that's why they can't make any meaningful choices, because they have no information to base those choices on. In turn, Chihuahuas rebutted these assertions by, among other things, citing evidence that generally plants' parts cannot survive on their own, that responses are coordinated between plant parts, e.g. root controlling shoot growth or vice versa. Relying upon communication between them, that memory, albeit not residing in the central repository, operates at a plant-wide level, and that plant decision-making goes well beyond merely achieving homostasias or making simple binary choices. Moreover, much more evidence of the complexity and coordination of plant behavior has been adduced over the years since Chihuahua's initial publication. Of special relevance to the study system that we discussed, Chihuahuas, rather than seeing modularity as an obstacle to intelligent behavior, suggests that a consequence of the repetitive modular structure is that the individual ramets might be regarded as being like parallel processors contributing to different experiences resulting from different ages to present-day decisions. This is going to be really fascinating because as we're going through this, my mind is applying everything that I'm listening to or that I'm reading to you as what's happening with no-name Dracaena. So this is a really useful conversation for me to think about this in the aspects of no-name Dracaena, of like, what does this mean? And I'm really kind of fascinating. Yeah, Veronica, I'm also curious as to what this is going to like, not only this paper, but exactly what's going to come out of this investigation that I'm going to, anyways, my own personal research is going to happen. I'm really excited about this. Okay. Moreover, much more evidence of the complexity and coordination of plant behavior. Okay, sorry, I just went backwards. A consequence of, that's where I was. I'm losing myself right now. Following up on this idea, we hypothesized that the additional level of modularity displayed by clonal plants that occur as networks of connected individuals could confer a greater capacity for intelligent behavior. So in this particular case, they think of it as a network of individual behaviors. So if we think about Pando in particular, it's really easy to think about this because you have each individual trunk that comes up. And so if you think about each one of these trunks as an individual, or if you think about a potato, each tuber that puts up a whole series of leaves, even though they might come from, because they're clonals, but those are considered the individual. In the name of no-name Dracaena, I ended up dividing based on, there was like a pieces that were all like the leaves were kind of open, but they all looked sort of together. They felt together. And then there was this one lone one that came up. But it still opens the question of, at what point do they become individuals? Is it because I considered them and I broke them apart as an individual? Or actually were they already individuals inside of? And so now as I'm thinking about other Dracaenas that I have, other snake plants that I have in my home, I'm picturing them because I just spend a lot of time looking at all the plants that live with me, and I can understand that maybe there are already individuals in there and they're choosing to act as a connected one. That's the reason why they're happily all in the same pot. And that when I get a message that says separate me, it's not because of only of space, because I have gotten that same commentary even when it wasn't a space thing, but very much I'm ready. Ooh, this is interesting to think about. Okay, I'm gonna be thinking about a lot of these topics. Okay, here we are. Now we're all the way down. The plants in such a network would benefit not only from the varied experiences of the constituent individuals, but also from modes of communication coordination that rely upon physical connections, and which could complement those modes that are also available to fully separate individuals. Additionally, we hypothesize that epigenetic modifications, which have already been suggested as a plant memory mechanism, that's what Galeano was talking about the fact that those epigenetic changes are the way that the plant remembers things, may be especially important in clonal plants, potentially contributing further to heightened capacity for intelligent behavior. Here we present these hypotheses against an overview of recent advances, elucidating plant learning and communication capabilities, and then propose particular research approaches to ascertain whether epigenetic modification does in fact contribute to adaptive behavior in clonal plants. Finally, we briefly explore the possible role of the potential associated fitness advantage of heightened individual intelligence in the proliferation of clonality in plants. So this is what we're going to be looking into. It's going to be good. So Michelle says, an ancestor says that they are the same until they're not. And I get the feeling that this is an us problem and not something he worries about. That is, I can totally feel into that. I can totally agree with that. That is a very interesting perspective. It's almost like, I don't know. I was thinking about this from the concept of, I think we get stuck in human definitions because of, again, our strong individuality. But I was thinking about this in reference to Siamese twins. I have to admit that I have watched a few specials that follow certain Siamese twins. And I think that there is something very similar in that, yes, they are two individuals. They very much are, but especially Siamese twins that share a certain amount of body perspectives, like a lot of their body is shared. I have also heard from them saying that, yes, while they are individuals, there are certain aspects of them that are kind of the same. And I think that that's probably the closest approximation that we have to think about this. And that also, if it wasn't for the fact that we as humans have such a strong need for our individuality, which I think is an error on our parts, it's kind of like a baby and a mom, like while the baby is still in the body, in the womb, I feel like there is a part that is like, there is an individual growing out there, but at the same time, it's still very much just all me. But we have this need of feeling this human individuality and giving people this extreme individuality that we don't allow ourselves to enjoy the sameness or the fact of union of that level. So yeah, I think you're right, Michelle. It feels like there's, and thank you to the ancestor who shared this with you. I do feel like this is a human problem. This is us. Okay, so remembering and learning from environmental interactions. There have been ample demonstrations of plant abilities to not only store information about their environment interactions, even for several generations, but also properly evaluate that information in the context of ambient conditions. Moreover, plants have even been shown to predict future conditions based upon past experiences. We've seen this multiple times, right? Thus, plants can perform quantum computing in chloroplasts to anticipate future light quality changes based on former light quality, enabling them to adjust photosynthetic apparatus accordingly. Similarly, plants can also modify their growth in accordance with expected future resources competition based on previous experience. By the way, I really love the fact that there's quantum computing in chloroplast. I do think that I love when we start to see terminology that we think maybe be relegated to certain kinds of thinking put into our scientific thinking, because I think it helps to bridge the gap between these. Finally, memories of previous environmental stress can also help plants better overcome future stresses. Learning from past experiences requires memory. Although some memory mechanisms, such as the accumulation or stable modifications of key regulatory proteins, cannot be separated from developmental processes, most plant memories are likely enabled by molecular mechanisms, of which epigenetic modification is probably the most prominent. Epigenetic mechanisms such as histone modifications and reversible methylation of cytosine alter gene expression and thus also phenotypes. Because epigenetic variation is environmentally inducing and mediates phenotypic plasticity, it is very likely that many of the environmental interactions of an individual are reflected in epigenetic changes. Which makes perfect sense, right? This is all just sort of stating the obvious to make it very clear, but that is it, right? You have these gene expressions that are changing. We've talked about this as inside of Reconnect and Plant with the Plant Kingdom. The course, it talks about these types of changes and the fact that plants are able to alter their gene expression. And so therefore their phenotypic responses, the way that their genes express themselves through this changes over time, even if the gene itself is the same. So these kind of variations makes it obviously is probably part of where the memory is. It's in these modifications of all these different individual items. Environmentally induced epigenetic change can become fixed for a period and eventually may become heritable, thus altering plant environment interactions in subsequent generations, right? And this is an important aspect of how plants share information from one generation to the other. Remember, we talked about that for us as humans. We think of it, we're kind of stuck a little bit in the idea of looking at one lifetime if you really are connected to your whole past life experience, you might be able to prepare yourself for what you're going to need in the future life. But it's not very much something, even in spiritual circles, it's still not something we really talk about. Plants do this relating to their seeds and everything. They put everything that they need in order for the next generation to be able to, pick up from this piece. And I think that this is looking at that using the clonal mechanism could be a great way to sort of better prove this, so that then we can go and look at it from other mechanisms as well. Let's see. Epigenetic variation can thus enable plants to remember their former interactions with the environment, learn from them, and apply the acquired knowledge to improve their performance in the future. Moreover, such behavior, because it can be performed at any stage of plant development, is clearly independent of developmental progressions, and thus overcomes the objection that suppose the behavior is actually just development, and therefore not a manifestation of intelligence. Ooh, I like that. I like the way that they're explaining that. That's a great way of really stepping into it. Because what they're doing here is they're using this example to go through what Fern and what others had talked about, which again, we've talked about a lot of these papers, the rebuttals to the idea of intelligence. And they're going through and they're saying, no, look, but this is a really great example that sort of shows that we're not just talking about like plants slowly progressing through, or developmental progression is the idea that it's like these tiny little changes that are happening almost without noticing them, kind of like, I don't know, your hair turning to gray is a developmental progression. It's not really an intelligence. My hair, I don't actually know. It's a whole series of processes that are happening chemically in my body. I can't control them, nor do I choose them. They just happen on me. Versus in this particular case, they're trying to say no, that you're looking at this type of stage of plant development, and that pretty much proves or can prove the idea that this is actually an intelligent choice that is then being carried out through the organism. Very, very smart way of looking at it. The potential for clonality and epigenetics to combine in facilitating plant intelligence. Clonal reproduction can yield genets comprising of up to several hundred connected ramets. Each ramet essentially constitutes an individual plant, generally capable of persisting independently if it becomes disconnected from the rest of the genet, spontaneously or by injury. However, the interconnected genet functions in some ways as a single entity, with stressed ramets able to avert death by drawing on the supports of the stronger ramets. So, right, this is what they're talking about, how the way that this grows in this clonal quality is you have this genet that grows out and then this ramet that grows up from there, right? And so what we're looking at is like the individual, if you're looking at the quaking aspens, for example, the individual tree is independent and can live on their own if I was to cut that tree off and put that tree in another location. But when that tree is connected in and something happens above ground, they have the safety net of what's happening underground in this giant genet that they can pull from. So in other words, other of the ramets, other of the trunks will then, use and be able to share information and nutrition, nutrients and things like that in order to happen. Let's see, labor division among ramets, and also with the overall growth of a genet, increased by its capability to share resources among ramets. Genets also have the ability to adjust to resource heterogeneity by placing ramets in favorable patches, showing that they take account of spatial environmental variation, which is especially likely to be encountered as they spread. So this is important, right? They move in and they grow in the direction of where there is the best and the most favorable conditions for the ramet that's eventually going to come up. That's pretty intelligent, right? To know where to move towards. It's kind of like human beings moving to a better neighborhood because they know they're going to have kids and they want to have a better school system. It's that same kind of thing. Additionally, because a genetic typically produces new connected ramets over time, such that there is a coexistence of ramets of different ages, the ramets within the genet are likely to have been produced not only over spatially varying environmental conditions, but also over temporally varying conditions. If information on such variation can be stored, shared, and translated into appropriate resources, then this can form the basis for particularly intelligent behavior on the part of the clonal plants. Of course, because now they have all this extra information that they can pull from. Spatially, they can assess a greater area in order to know what's happening, but also temporally. So one older plant could say what's been happening there over time, and they can share that information down to a younger part that is coming up. So I think that that's really an important aspect to remember, that now the plant has like double the information, or triple or quadruple, depending on how many ramets come up. Clonal plants do appear to have greater ability than non-clonal plants to remember past environmental interactions. This may be due, at least in part, because of their more effective transfer of epigenetic information among clonal generations. And in contrast to the resetting of most environmentally induced epigenetic information during meiosis, during sexual reproduction. In contrast to the resetting of most environmentally induced epigenetic information during meiosis, during sexual reproduction. So this is basically saying that because it's a clone, it's more likely that they have all of the information, rather than in the sense of sexual reproduction, where you have two plants that could pass information that goes to the seed. This sometimes has to cancel out what has been shared, because they can't share everything, because some things might be, in other words, if one plant was growing up in one spatial area and another one in another spatial area, they might have had epigenetic information, in other words, gene expression, in different directions that might even contradict each other. So those would get shut off when they pass off. I hadn't thought about that. That's a really interesting perspective. It would be interesting to see how we move it to I have to think about this, but this is really interesting. In fact, the most faithful intergenerational transmission of epigenetic information would provide an advantage to ramets relative to sexually produced individuals, even if they become detached from their parent clone or are largely independent. Of course, because they've already had all of that information come from the genet that's been passed on to them. So the ramet that's come up brings with it all the history and all that. So it is the most faithful inheritance that they could potentially have. I had never thought about this before. Nevertheless, it is within connected genets that epigenetic memory is likely to have its greatest effect in terms of facilitating intelligent behavior of clonal plants. This is because although even productions of non-clonal plants can communicate via root exudates or volatiles, the physical connection maintained within such genets allow communication by additional means. Communication within interconnected ramets have been demonstrated, particularly with regard to early warning systems in which before we even get there, think about this also from the perspective of anything that one part of the clonal colony is learning. Because even if you have something as large as Pando, right, big system of quaking aspens taking up large football fields, and I like it because I can say football fields and it doesn't matter whether we're talking about like American football or we're talking about traditional football, aka soccer. It's all football fields. It's all huge, and you can just imagine it. But when I look at quaking aspens of Pando, right, large football fields, if they are still all connected by the same genet, then even if you have one part of the colony that is having and experiencing something, that information is being passed on all the way through to the newest members that could be on the other side. This is fascinating. This is because even though populations of non-clonal plants can communicate via root exudates, like I was saying, the physical connections maintained allows them to communicate by additional means. Communication within interconnected ramets have been demonstrated, particularly with regards to early warning systems in the information of a herbivore attack can be spread throughout the clone, enabling those ramets not yet attacked to produce defenses. Hormones such as jasmonic and salicylic acids are thought to carry each such signals within a clonal network. Although they can provide information advantages for clonal plants, these messengers would carry information only about the situation currently or very recently encountered by the genet, and would not convey information about events or conditions from the more distant past. No, because they've already passed that on when that. So you're saying, yeah, because they would have already passed that on. So the ramet that comes up would have the latest information based on up at the time that the ramet comes up. Then from then on, it has information about ongoing information. It doesn't have any need for the past because it's already acquired all of the past when that ramet first started to come up. Based on studies by Raj et al. And Richards et al. Showing that clonal genetically homogeneous populations of Populus and Fallopia, Populus, by the way, is aspens, I believe, then Fallopia, which I don't know, respectively remember former environments via epigenetic variation. We propose that environmentally induced epigenetic change might be the mechanism that can carry precise information about environmental interactions from the more distant past. We suggest that given the absence of meiotic resetting of epigenetic information in clonal reproductions, right? So yeah, nothing gets reset from one place to the other. In clonal reproduction, newly emerging offspring ramets can inherit epigenetic information from previous environmental interactions from the maternal ramet. Moreover, because an offspring ramet can encounter different environments than those experienced by the maternal ramet, the epigenetic information that will ultimately be conveyed by the offspring ramet to the generation subsequent to it, i.e. the third generation, can represent the combination of maternal, first generation, and offspring, second generation environmental. Yeah, basically, we keep acquiring more information and I pass all that information on. It would be interesting to see, based on the studies that Monica Galeano had about how long memory can be held by certain kinds of plants. I don't know if that changes based on the type of plants, because she only did her test on Mimosa pertica. But it would be interesting to see if that memory, how long does that hold? And therefore, does every generation get passed on? Or is there a point of decaying where some of the memories get lost and therefore, or maybe are no longer relevant and they don't have to be passed on? This would be an interesting piece. Indeed, from studies on Arabidopsis thaliana, we know that a single genotype can have more than 250 distinct stable epigenetic states. Maybe this is answering my question. Thus, in a genet consisting of tens or hundreds of interconnected ramets, each ramet is likely to have a different epigenetic state depending not only upon the environmental conditions experienced by itself and its progenitors, but also upon the interactions between these conditions and the phenotypic types in each of these generations. Hold on, I got to read that again. Depending not only on the environmental conditions experienced by itself and its progenitors, but also upon the interactions between these conditions and the phenotypes in each of these generations. So it's not only based on what you have experienced and what you inherited, but also on how these two things are interacting, which is the whole decision making. I not only take information and have like a plus minus on how I react to things, but I have an understanding, a digestion of that information that I have to have in order for me to decide which of these to apply to any given situation. That is much more complex intelligence than any other paper I have seen. Well, that's not true, but most of the other papers that I have seen. This variation among connected individuals, each of which consists of a whole, potentially independent plant, in combination with the enhanced capability for communication among them, would, we suggest, provide the basis for swarm intelligence, an emergent property of cooperating groups that enables them to solve problems that are beyond the abilities of a single member. Yes, that would make sense. So the idea that, of course, at this point now, you have all of these that share all these memory pieces, and therefore that allows me to have an intelligence that comes from the entire group rather than from any individual. Indeed, in seeking to explain the decentralized intelligence exhibited by individual plants, suggested looking at other decentralized intelligence systems, such as swarm intelligence, those found in social insects, such as ants and flocks of birds and schools of fish and such like that. This analogy is ever more appropriate when considering the potential swarm intelligence of ramets within a connected genet. I wonder if that would make clonal plants of this size a superorganism, or any plant that multiplies over itself. I mean, or are all plants, because of their decentralized nature, be considered a superorganism by the definitions used in things like biomimicry and I'm going to need to do a lot of thinking after this paper. Hold on, let's see where we are just from a time check. I have to go through all this, and then there's the conclusion. So the conclusion is on page 5, and we are going into page 4. We're going into. Okay, so you all let me know if you think I should keep going and just we'll go all the way through this. Yeah, coral reef is another one. Good one, Veronica. Really nice. Yeah, that a lot of coral reefs act in that way, as that decentralized superorganism. So you all let me know if I should keep going. I have about another page and a half to do. I can comment a little bit less so that we can get through it, or well, let's see how long it takes. Although we are primarily concerned with a relative capacity for intelligent behavior of a connected genet versus an array of non-clonal, yet comparable individuals versus an array of non-clonal yet comparable individuals, the ability for constituent ramets to process information. Hold on, I just had something pop up, excuse me, that popped up on me, and I wanted to double check that I am not missing. No, nothing. I wanted to just double check that I didn't have another call after this or anything like that, but I have time. To act somewhat anonymously does raise the question, with possible evolutionary implications, of whether a genet would have an intelligence advantage over a single plant of similar size to the entire genet. In more fully considering this possibility, which is beyond the scope of this paper, an important issue would be whether the possible communication advantages enabled by greater physical interaction within a single large individual would offset the benefit of having a swarm of largely autonomous ramets. It's a really good question. It's a really good question. This is a really good question of, like, comparatively speaking, from a human perspective, is like, is one, or maybe, one really intelligent person have an advantage when they're working through a problem than a group of pretty smart people working on the same problem? Which has the advantage? The one that has all of this giant, like, genius level intelligence type of thing and knowledge and information, and can process things based on all their multiple experiences, but it's all still confined into one, or the diversity of the entire group of people that are collaborating on it all together? There's a question for you to think about. One possible product of epigenetically mediated intelligence in clonal plants is pre-programming of offspring ramet phenotypes for particular types of growth based on previous interactions of the genet with that environment. That's what I had said. We as humans, we might be able to think and conceptualize our past lives, but we very rarely think about programming for our future life. This is something at Damanhur I learned, because of course we program for the future too. And we think about, like, children coming into the community, and we try to help kind of program and work out what are the best characteristics that they could possibly have based on the environment. And so this is a really interesting perspective in that. This can significantly increase overall success of the genet, particularly in cases where environmental conditions are predictable. For instance, adaptive responses to drought may encompass increased investment in roots to better tap groundwater and/or reduce leaf growth to reduce water loss. In the case of simply phenotypic plasticity, the newly emerging offspring ramet would respond to the ambient water level and adjust its growth accordingly. Thus, in the scenario in which rainfall temporarily moistens soil of an otherwise dry environment, a newly emerging ramet would adjust its phenotype to wet conditions and grow shallow roots and large leaves, which could easily become inefficient or even costly for the genet when the moisture disappears. However, knowledge of previous acquired by the genet could theoretically modify the plastic responses of such a new ramet to take better account of the long-term environmental conditions. Very interesting, very, very interesting about how they pass this information on and use it. Such intergenerational phenotype adjustments are well documented for sexual generations, and we propose that similar effects are likely to occur in clonal plants. Of course, it is possible that the constraints imposed on plastic responses to contemporaneous conditions could be detrimental if they reflect conditions that are no longer at all relevant. Nevertheless, we suggest that in the ongoing communication among ramets from differing periods may allow, via swarm intelligence, the genet to evaluate the suitability of differing responses and then translate this through signaling and response allocation into decisions to favorable outcomes of constituent ramets. It's almost as if the machine is more like the intelligence, the mind is more alive. The mind is more able to react accordingly and more on the spot, because there is much more, because there's this clonal property, as opposed to an individual that sort of constrains to the memories of only what that individual received from the very beginning of time. I think this also connects very much to the size of the individual, might have something to do with it. Something to consider. Indirect support for this idea may have been provided by Luap, who and all, who in an experiment on potentilla and Potentilla anserina found, I think that's what that is, P is probably for Potentilla, found that as the stolen lengthened, the decision about where to place successive rooting ramets depended not only on resource richness experienced by the newest ramet, but also by the preceding year. Additionally, the placement of new Potentilla reptans ramets was influenced by the variability in richness among the rooting sites of previous ramets. So basically, this is just proving the point that we talked about before, that it's not just the experience of the individual and what conditions they are. So it's not like they're given a set of information and then they use that information only based on where they are, but they, in this particular case, the ramet can keep feeding them information. And so they have a bunch of information from multiple generations in order to make better decisions. Thus, for this species, their model system demonstrated that not only the average amount of resources obtained by older ramets, but also the variability of resources among ramets can alter the growing strategy of a whole genet. Although they considered only plant hormones or resource molecules as potential messengers in their system, epigenetic variation can also underlie the behavior of the clones in the study. Variation among epigenetic memories of interconnected ramets can also modify the information values of signals that are sent within the genet, because epigenetic variation alters plant responses to hormones and other messengers. Potentially, each ramet in a genet could respond differently to signals behave. Of course, because each ramet is still taking in information from where they're currently located, so they receive the information, but they react to it different. It is also possible that epigenetic distinct ramets can differently modulate the intensity of signals that they are sending. Thus, the interaction of interconnected ramets can form the basis for a unique system in which the information spread is evaluated and modulated in the form of epigenetic memory. I was just thinking about how I wonder how, if you could compare the idea of take a forest floor, where even if you're talking about multiple trees of the same species, so full information share, but they have to go through a transmitter like the mycelial network, some information gets lost because the mycelial is not able to carry all the information necessary, as opposed to, in the case of a clonal species, the genet can pass everything because, of course, it's all coming from the same species. So this is really interesting. For all the above mentioned reasons, we suggest that epigenetic memory within connected genets can enable information storage and evaluation such that it can provide the basis for variability in decision-making that would overcome objections. That plant decision-making is trivial. It would overcome objections that plant decision-making is trivial, thus not manifesting intelligence. Moreover, such variability could enable genetically and even developmentally identical genets to react differently to environmental stimuli, such as attack by insect or microbial information. All right, accessing intelligent behavior of clonal plants. Although we do not possess a test to quantify the overall intelligence of clonal plants, all right. All right. Bye-bye, Veronica. We'll catch up with you afterwards. And yeah, I'm going to keep going because there's only one more page and I think we're good. Although we do not propose a test to qualify the overall intelligence of clonal plants, i.e. something akin to an IQ, we believe that the extent to which information is stored epigenetically transmits across space and time within connected genets, and ultimately incorporated into decision-making of constituted ramets should be assessed. Thus, conceptually, to test whether epigenetic conveyed information from the past is useful to the plants in making better decisions, i.e. that will yield greater fitness or proxy, therefore, we can compare two genetically identical and developmentally equivalent genets, one naive, i.e. without such information, and one processing such information about past environments, interactions. If the plant's behaviors were guided only by presently sensed information without input from acquired memory, then both genets should react similarly to present environmental stimuli. However, if the plants have the ability to evaluate new ambient signals against the backdrop of stored environmental information shared through clonal network connections, the genet with relevant experience should behave differently, giving it a considerable advantage over the naive one. Yeah, I think in this case, what they're talking about also is that you have to let it grow so that you have two ramets that receive the same information. Then you have to split them. One stays connected to the genet, the other one instead into another environment. I think this would be the best test. And then if they're both the new ramets are presented with the same stimuli, do they react similarly because they both have the same information based, or do they react differently because also the genet is continuously passing new information? So you would have to have the genet have had an experience that then changes the way the reaction would be. These are a really interesting test. And I think that this is already starting to show our relationships and how the relationships are different. How can you not think plants are intelligent after all this? To specifically examine the role of epigenetic modifications in furnishing such memory for decision-making by clonal plants, perhaps the most straightforward way to make this comparison would be to use a demethyling agent that would disrupt already acquired epigenetic modifications. Oh, I hate when they do these experiments in this way, because it's like destroying something. But using this approach, equivalent genets would be exposed to the same sequence of temporally varied environmental conditions, but for one set, their epigenetic memory would be disrupted continuously via DNA demethylation using a demethylating agent such as whatever. Demethylation can be successfully applied in studies testing the role of epigenetic variation and phenotypic blah, blah, blah. Hold on, let me see where we get into the interesting part. These are all the experiments. In other words, we have developed a new approach in which mature clonal plants are sprayed repeatedly. Leaves, nutrients. Okay. So basically what they're doing is that they're exposing the plants to a series of agents in which the plants that have been separated out, so that the offspring ramets have a different pattern. Okay, hold on a second. Create arguments such as an approach that can serve a last of proof of principle. Okay. Without significant effect on other, although this method may have side effects, can create. Okay. Basically, this is a way that they're trying to separate out whether or not what is the type of information that the genet is still passing on to the ramet. So there might be modifications to see whether or not this then over time still continues to pass on phenotypic responses, which is what we've talked about all along. A more sophisticated and complicated approach would be to grow genetically identical, developmentally equivalent genets and subject them to innocuous variations in environmental conditions that could serve as a signal for subsequent more consequential environmental changes, i.e. equivalents to such signals used for conditioning, okay? By pervading such genets with various environmental signals and consequences, as an example of varying herbivore intensity, and then seeing whether past experiences favorably alters responses to present conditions. We can assess whether genets are learning from experience and applying this knowledge. Exactly, that's what I'm saying. So you're really just testing to see whether or not the genet itself is always pumping new information off. The latter approach can be combined with experimental demethylation to determine the mechanistic role of epigenetic modification. Yeah, you don't have to do that. You don't have to hurt them. Just trust that it's happening. Additionally, to examine the extent to which information is shared and has impacts across a genet, individual ramets can be treated differently. Exactly. An assessment that can be made of whether or how much their neighbors show responses over a longer term that would be accounted for by hormones. Similarly, although ramets of different developmental stages may respond differently to particular environmental conditions, effects on neighbors of those at the sensitive stages can be assessed to determine whether a ramet can learn from connected ramets experience. Finally, the extent to which epigenetic variation within a genet contributes to variation among ramets in response to an environmental stimuli can be assessed again by demethylation, which by eliminating some of the epigenetic variation can convert. So I'm going to stop because this is kind of like, it gets really technical. Basically, what they're looking at is what are all the different ways in which I can test whether or not like genet, so the underlying structure that connects everything, all these different clonal ramets together, right? So if I have, how do I make, what modifications do I make in the genet that actually get passed on to the ramet, both at the stage of creation as well as once the ramet is up. So that's one piece. And then the other thing is when I have two ramets, what are the changes that I can make between ramets that will be, how will the plant, how will that ramet respond based on what is happening? So these are all the different test scenarios. It's not really important to understand the actual test itself as much as for what are the different testing scenarios, because what they're doing is that they're suggesting these approaches because they say, we're going to see, they predict that based on all this, we're actually going to see an increasing complexity of the way that the plants respond. So because they are proposing, additionally, because we propose that intelligence in clonal plants would arise from multiple factors, including intergenerational transmission, epigenetic modification, and ongoing enhanced communication. So two things. One is the fact that all of the past comes in when the ramet starts to come up because the genet passes everything through. And then there's an ongoing communication that's much faster through physical connections among the ramets themselves. So as the ramets are experiencing new things, they're sharing that down the genet, and that's coming out to all the ramets. So there is this constant flow of information. That means I'm taking in information here and I am passing it on to over there. And over there is then looking, how do I apply that information based on what's happening? And so they're recommending a whole series of different modified tests that can be done to see how much of this is happening, confirm that it's happening, and then also show whether or not this is much more powerful than see 3 or 4 plants that maybe grow in 3 or 4 trees, for example, just using a tree as an example, that are growing in a similar area from the same parents that created the seeds, and then they're growing in different areas, but their communications are often all happening, if at all, through mycelial networks and through other kinds of mechanisms. So they're not as complex or as robust as would happen with clonal plants that are still all connected. Thus, perhaps comparison can be made between intact clonal networks and networks with connections severed after a network has already been subjected to varying environments. Or less intrusively, I like that they're talking about less intrusively, similar comparisons could be done between networks and equivalent numbers of single ramets in the same species. Additionally, to discuss and address the question of clonal versus non-clonal intelligence, the ability of connected genets to respond to environmental variation can be compared with that of arrays of similar and phylogenetically close non-clonal individuals. Although all of these comparisons would have to overcome challenges, we believe that it is worthwhile to at least suggest such possible avenues for further research. So that's what I was saying. These are all suggestions that they're making for what ways we can study this further so that we can better understand the complexity gains that clonal species have because of their connected network. Which makes a lot of sense from one perspective, right? It's the whole idea that because I'm so interconnected, we can extrapolate this out to a human perspective that says we are much more intelligent. And Dominher is a great example of this, right? The fact that we're a community, that we're constantly sharing information, the fact that even if we're in this particular case, obviously not all clones of one another, but the fact that there's a constant flow means that we're actually much more, gives us the ability to be much more complex and much more, and allow the intelligence of our future generations to be based on experiences happening with the past generation, but then they use their current intelligence and their current experience to kind of mix these two together. So you get the best of both worlds. So it's kind of like saying that any kind of family or united network of any sort, right, is always better when there is a constant flowing of information, both from inheritance, so what I pass on to the next generation. So this is the whole idea of what is the legacy that you leave behind and how readily accessible is it to somebody else to take up this legacy. I have several of my clients that are at the legacy stage of their careers or their lives, and that's exactly what they're looking at. What is the legacy that I leave behind in order for it to be inherited by the next generation? And how does that next generation use that to go forward? So it has to be consumable, something that they can integrate into their lives. And I think that even if you just take this out of the context of a clonal quality, what it's also helping us better understand is the importance of this, because that's the way that we're not going to we pass on to a new generation of information that then they apply in the way that they see best. So it's not a you must do this, but more of here are all the conditions that led me to making a certain definition or a certain decision, and therefore you then take in what's happening now, look at how we interpreted it, but then make your own interpretations based on what's happening today. And I think that this is where a paper like this really helps us think about it from the perspective of how does the human learn from this model that plants are providing for us. Tarawa suggests that intelligence must be understood in terms of the interaction of the individual with the environment, and the environment posing the problems that the intelligence is needed to solve. As examples from the literature show, plants are able to draw lessons from their interactions with the environment and can respond in ways that cannot be characterized as simple physiological or developmental consequences. Moreover, we suggest that clonal networks, especially in conjunction with epigenetic modifications, can facilitate intelligent behavior, with epigenetics enabling long-term information storage in response to the environment, and the latter permitting not only unaltered transmission. This is the first time they say this, but I think this is really big. Not only unaltered transmission of this information to subsequent generations, but also integration of information across multiple units experiencing different conditions. These two elements right here, in my opinion, are extremely important. Unaltered transmission. So how do I provide this information to the next generation in the way that it is most pure and clear and does not have to be altered in its input? So it doesn't, this is the difference between a passing things on from word of mouth, where it is always the transmission is altered based on the person that is passing it on, like that who received it and then passed it on. In this case, unaltered transmission of this information to subsequent, and also then the integration. So how consumable is it? How easy it is for me to take it in unaltered, and how easy it is for me to consume and to bring it in and to take in from the multiple experiences. A longstanding question in plant ecology has been why the clonal lifestyle, despite some likely disadvantages in terms of reduced population genetic variation, is so widespread and indeed predominant in some habitats. The position facilitation of intelligent behavior, if actually present, might at least in part explain why the success of clonal species, by enabling them to have more flexible, accurate, and efficient responses to varied environments that then do non-clonal species. Not only might clonal plants gain an advantage over non-clonal plants, but it is likely that there are differences among clonal species in their abilities to accumulate and or employ stored information, i.e. their intelligence. In particular, their intelligence could depend on life history characteristics, such as clonal growth type, integration of level ramets, the level of information exchange among the ramets, and persistence of clonal connections. Results from our first, as yet unpublished, studies on these subjects have shown that memories of former environments can persist across several cohorts of ramets and can be erased by demethylation. Because the research that our group can conduct is finite, and the questions, especially regarding potential advantages that enhanced intelligence can provide to clonal versus non-clonal plants, would require major undertakings to definitely answer, we hope that this essay will stimulate discussion about our ideas and possibly inspire other researchers to help to determine their overall validity. I found this to be really, really fascinating, very well written, clear, and very relatable in a lot of ways. So I'm very excited. So this is epigenetic memory as a basis for intelligent behavior in clonal plants by the authors here, which have names that not all of them I can, well, that's not true. Vitlatzel, Alejandra Rendina Gonzalez, and Jonathan Rosenthal. So I found this to be really, really interesting. I would love to hear, I hope that this gives you some more food for thought, and I hope it also provides some more opportunities for you to think through the different ways that, this is really fascinating. I think there's a lot of applications here that we can think about and extrapolating out models. Remember, as I said at the very beginning, a lot of it is also about like, what are the models that plants are affording us that are giving us the opportunity to look into? And I think that there are various models here that help us see that being really connected and really uniting and even receiving the same information doesn't mean I become a little copy, but instead gives me greater information necessary for me to adapt that based on the conditions that I'm experiencing right now. So, but I look forward to hearing, your own thoughts, to reading your thoughts. So please feel free to leave a comment. If you're watching the replay, thank you so much for, being here and for experiencing this. And I really look forward to your comments. Remember, next week, we will not be having actually our commentary, because next week we will be having our final book club discussion on Thus Spoke the Plant by Monica Galliano. So when we do the final discussion, we actually do not have live commentary because it is impossible for me to sit through that many hours one after the other. But we will be back on the 24th of November. Even though that is a holiday weekend, I I am thinking I'm still going to do a live session, but I'll let you know if anything changes. Anyway, I look forward to all of your comments. Thank you so much for being here and appreciating and being part of this discussion. And until we will see each other next time, and I look forward to your comments. Bye. This is Tivia. Remember, resist the urge to hold back your evolving queen brilliance. And we will talk. Bye.